Good afternoon, CGIL. This week, we are happy to have Dr. Aryn Wilder, a conservation geneticist at the San Diego Zoo Wildlife Alliance. Dr. Aryn Wilder applies genomic tools to study the impacts of human activities on natural populations. Her work focuses on identifying populations at risk of extinction and developing effective strategies to conserve them. Dr. Wilder's research spans topics such as rapid evolution driven by harvesting or habitat fragmentation, the spread of introduced pathogens, fitness trade-offs of inbreeding and outbreeding, and the role of genome-wide genetic variation in extinction risk. With a PhD in biology from Boston University, Dr. Wilder has held research positions at leading institutions, including postdoctoral roles at Cornell University and Boston University. Since 2018, she has also served as a visiting scholar in the Department of Biology at the University of California, San Diego. And Dr. Wilder is dedicated to integrating innovative genetic research with on-the-ground conservation efforts to protect biodiversity for future generations. Today, Dr. Wilder's presentation will be on using genomics for the conservation and restoration of endangered species. So I'll turn it over to you now, Dr. Wilder.
Great. Thanks so much, Gabby, for that introduction, and thanks everyone for the invitation to come and give a seminar. This is really a great opportunity and honor, and so thanks again for the invitation. So as Gabby mentioned, I am a scientist in the Conservation Genetics Group at San Diego Zoo Wildlife Alliance. So today I'm going to talk about how we are using genomics for the conservation and restoration of endangered species. So we are in the midst of a sixth mass extinction marked by accelerated loss of biodiversity, population declines in more than 2/3 of vertebrate species, and 28% of species now threatened with extinction. And this figure here on the right shows the acceleration of extinction rates in a number of vertebrate groups over the past 100 years beyond the background level of extinction before that time period. But there's still reason to be hopeful. Conservation actions do help. And a recent study showed that in 2/3 of the cases analyzed, conservation actions had a positive impact on the biodiversity targets. So conservation is a major part of the mission of San Diego Zoo Wildlife Alliance. Our mission is to save species worldwide by uniting our expertise in wildlife care and conservation science with our dedication to inspiring passion for nature. So our conservation science projects are roughly organized into these geographic conservation hubs distributed around the world. And the San Diego Zoo Wildlife Alliance is an international nonprofit conservation organization with 2 front doors. Move this box out of the way. Okay. So we consider ourselves having 2 front doors: the San Diego Zoo, which is located in the city of San Diego in Balboa Park, and the San Diego Zoo Safari Park, which is north of the city in Escondido. And our conservation science and wildlife health staff, most of us are located in Escondido next to the Safari Park at the Beckman Center. The conservation science staff are a team of multidisciplinary scientists specializing in veterinary and wildlife care, conservation genetics, recovery, ecology, population sustainability, disease investigations, reproductive sciences, and community engagement. And we bring all of these areas of expertise into our conservation projects. So I'm part of the conservation genetics group, and within conservation genetics, we have further specializations into things like cell culture and biobanking, population genomics, landscape genomics, and genomic modeling. So today I'm going to talk about 3 cases that show the ways that we use genomics to improve our insights by leveraging information and coding genomes about a population's past, present, and future that are relevant for conservation. So the first is using reference genomes to assess conservation status and predict extinction risk from that encoded information, and that is in a study across mammalian species. The second is how we are using population genomics to inform management strategies for endangered populations in the Pacific pocket mouse. And then last, how we're using genomics to predict future viability and resiliency of recovering populations. So that is, I'm presenting the case of white rhinos. Okay, so jumping right in to the first example of using genomes to help predict extinction risk in mammals. With so many species in decline and threatened with extinction, we need tools for prioritizing species that are in need of conservation interventions and actions now. And one of the big sort of organizational tools that we have is the IUCN Red List of Threatened Species, which takes information about recent population trajectories, threats, species ecology, and distributions, and compiles all that information to categorize species by into these categories that tell you something about their risk of extinction. So categories range all the way from already extinct to critically endangered to near threatened and least concern. And in this list on the right, the numbers to the side of each category represents the number of species that are categorized into each of those. And you'll notice that the data deficient category still has about 20,000 species that are considered data deficient. So lacking adequate information to actually put them into one of these extinction risk categories. So with the growing number of reference genomes constantly being assembled and published, the idea is that we can leverage the information encoded in those genomes to help us assess extinction risk of species. So as I've mentioned, genomes hold information on a population's history as well as its current fitness today. And with the growing number of genomes available and increasingly sophisticated tools to analyze those genomes, we can actually access that information. And so one of those tools are multi-species genome alignments, like the Zoonomia alignment of mammalian genomes. And these alignments of whole genomes can help us to identify functionally important variation, genetic variation that's in the genome, as well as provide a framework for systematically comparing genetic variation across species. So then, our main question is, can reference genomes tell us something about a species extinction risk? And we used the Zoonomia multi-species alignment of 241 mammalian genomes to test this question. So across the alignment and the phylogeny created from the alignment, which includes one genome per species, we estimated variables of genetic diversity, so things like runs of homozygosity, genome-wide heterozygosity, and the distribution of heterozygosity across the genome. Historical effective population sizes of each species, and species genetic load, or the total sum of potentially harmful mutations in the genome, and compiled all this information into extinction risk models. So we used all this information to train and test these models. So we first estimated historical NE, so historical effective population size, using the program PSMC. So effective population size is a measure of the strength of genetic drift in a population. So it tells us something about how much genetic diversity the population can retain, as well as how efficient natural selection can act on that population. If genetic drift is really strong, then natural selection isn't as efficient, and the population can't retain as much genetic diversity. Effective population size tends to be correlated with census population size, but it's usually smaller than census population sizes. So on the right here, this figure shows the NE trajectories over time of all of the species in the zoonomia data set, arranged by their order, the phylogenetic order, and colored by the IUCN Red List status. And you may notice that the NE estimates go to as recent as 10,000 years ago. And that's because from a single genome, we can get good estimates of past effective population sizes, but estimates more recent than about 10,000 years ago are not reliable. So we only used NE estimates from more than 10,000 years in the past. We also estimated deleterious genetic load. So again, this is sort of the sum total of harmful mutations or potentially harmful mutations in the genome. And we did this first by identifying substitutions relative to the closest reconstructed ancestral sequence. So what that means is basically this cartoon here zooms in on a part of this phylogeny where we have these two sister species, A and B. We can compare changes like nucleotide substitutions relative to their ancestor. We can also identify heterozygous variants from by mapping resequencing data or short read data back to the reference genome to identify heterozygous variants. We then infer the impact of these substitutions and heterozygous variants using a few different methods. So one, by the degree of phylogenetic conservation of a site. So we assume here that any site in the genome that is really highly conserved across all mammals is probably under strong purifying selection, because it's really important that it, stays the way it is. And then any change to that site is more likely to be deleterious. We also assumed that changes to protein coding sequences were more likely to be deleterious than synonymous changes. And then we also sort of categorize the potential impact of a change in a given gene based on the impact of knocking that gene out of a mouse model. So if it has a lethal impact, then that gene is probably really important for viability. And then all of the statistical analyses for this study we ran in a phylogenetic context. So we used the phylogeny to account for non-independence in these in comparing across species. So our study showed that historical effective population size was the best predictor of contemporary conservation status of all of the genomic variables we looked at. So threatened species tended to have smaller historical effective population sizes, which is maybe not super shocking to anyone, except when you consider that these effective population size estimates were from more than 10,000 years in the past, which suggests that population history, even distant population history, is still informative of a species extinction risk today. For a subset of species that also had census population size, like contemporary census population size, we also looked at the NE-NC ratio and found that threatened species tended to have inflated NE-NC ratios, or another way to think of that is a smaller contemporary census population size relative to its historical effective population size. So that suggests that threatened species have smaller contemporary population sizes than expected from their historic effective population size. And the three species with the highest NE-NC ratios were bison, hirola, and giant panda, all of which were known to have declined from much larger historical effective population sizes, or historical population sizes in the recent past. Historical effective population size also determines the burden of genetic load that is fixed in a given species. So species with smaller effective population sizes had proportionally higher levels of deleterious genetic load among all of their substitutions. And this was true in both in genes that had lethal effects in knockout mice and also genes that had viable effects. So this is consistent with population genetic theory and provides empirical support for the accumulation and fixation of deleterious alleles in small populations. So we then modeled all of our genomic predictors of extinction risk, first summarizing a lot of our highly correlated variables with principal components, and then testing the relationship of the principal components with conservation status of species. And there were two principal components that popped out, PC1 and PC3. PC1 roughly captured genetic diversity and genetic load, and PC3 distinguished different types of genetic load. So in total, the models suggested that threatened species tended to have smaller affected population sizes, lower heterozygosity, more deleterious mutations in coding genes, and more deleterious alleles that were homozygous or fixed across these species. So effective population size was the strongest predictor of extinction risk. And we also found that its effect varied by context. So for example, an herbivore was more likely to be threatened by a small affected population size than a carnivore or an omnivore was, which supports previous findings of elevated extinction risk in herbivores despite their larger population sizes. And rodents and bats were also less likely to be threatened by a small NE than other orders. So then we combined all of this information that we had into machine learning models to predict threatened or non-threatened conservation status. So all of this information, this genomic information, included 13 genome-wide summary statistics, about 45,000 window-based metrics. So windows being windows of the genome shown here in rows, and comparing across species here in columns. So these window-based metrics of genetic diversity and genetic load, and then also included ecological variables from the Pantheria database. So ecological variables would be, for example, a species' body size, habitat breadth, generation length, or litter size. So of all of the models we looked at, models that had ecological variables were the best predictors with an AUROC of 0.88. So these AUROC metrics just sort of measure how often the model is right versus wrong. And ecological variable performance is not especially surprising that it's so good, given that the many of the ecological variables that went into this model overlap with the information actually used for the IUCN assessments. But we also found that genomic models were able to non-randomly distinguish threatened from non-threatened species, which suggests that these genomic models can be especially valuable when other data types are lacking. So for those data deficient species. So to demonstrate how these kinds of models might work, we applied them to 3 data deficient species in the Zoonomia data set. So those were the Upper Galilee Mountains blind mole rat, for which we had no ecological data available from the Pantheria database. The killer whale, surprisingly, is data deficient and has no IUCN status, and the Java lesser chevrotain. So across all of the different models that we built, the killer whale came up as having the highest probability of being threatened, whereas the Upper Galilee Mountains blind mole rat came up as having the lowest probability of being threatened. So if we were to take this information and this list of 3 species, we would prioritize the killer whale for additional follow-up studies and assessments. So to conclude, this study demonstrates the potential for using even single reference genomes to help us triage species for further studies and conservation assessments. And we expect that with the additional number of genomes that are coming online every day, along with their increased quality and the availability of resequenced genomes, we expect that these models will continue to improve over time. Okay, so now the next sort of case study I'm going to present is how we are using population genomics to help guide conservation of the endangered Pacific pocket mouse. So as I touched on earlier in the talk, across the globe, most populations are getting smaller. And in the past 50 years or so, on average, populations have declined by about 69%, which increases the number of populations that are vulnerable to extinction. And one of the reasons that small populations are vulnerable to extinction is because of this so-called extinction vortex, where you have some external factor like habitat loss and degraded habitat quality that causes population declines. And small populations have strong genetic drift, which causes them to lose genetic diversity. High rates of inbreeding lead to low survival and reproduction, which further reduces population size and causes this feedback loop that eventually ends in extinction. I just want to point out here that that this idea of increased levels of inbreeding in small populations leading to low survival and reproduction is what we call inbreeding depression. So that's when you have two individuals that are that have related parents. They tend to have more deleterious alleles in a homozygous state, so their survival and reproduction is lowered. So that's called inbreeding depression. So one tool that we have to pull species out of this extinction vortex is called genetic rescue. So if we take this really simple diagram where we have this small isolated population shown in orange that has low fitness because it's suffering from inbreeding depression. If we can facilitate gene flow from a larger outside population into this small population, then that influx of novel alleles can mask those homozygous deleterious alleles that cause inbreeding depression, increase overall genetic diversity, and boost fitness of this population. And that is called genetic rescue. And there are lots of examples of successful genetic rescue across taxa. But there are also potential risks of genetic rescue if things like genetic incompatibilities or local adaptations that distinguish this orange and blue population actually lead to reductions in fitness from gene flow between them. And when that happens, it's called outbreeding depression. And an extreme example of outbreeding depression is like the case of the mule, which is a cross between a horse and a donkey. Several chromosomal rearrangements cause sterility of the hybrid offspring. Fortunately, outbreeding depression is pretty predictable, and there are established guidelines to help us avoid outbreeding depression. So genetic rescue should not be, it's advised from these guidelines that genetic rescue should not be attempted when the source and receiver population have been isolated for more than 500 years, live in distinct environments, or have fixed chromosomal differences. But with so many populations in decline, then we can expect an increasing number of populations that may not meet this broad criteria. So genomic tools can help us to weigh the trade-offs and risks of inbreeding depression and outbreeding depression so that we can more precisely apply genetic rescue to manage diversity and fitness of small populations. And this is the goal of our work on the Pacific pocket mouse. So the Pacific pocket mouse was previously distributed along the coast of Southern California from as far north as Los Angeles down to the US-Mexico border. And Widespread development along the coast really accelerated in the 1930s. And after that, a lot of the populations along the coast became extirpated. And the species was actually thought to be extinct for several decades before it was rediscovered and emergency listed under the U.S. Endangered Species Act. And the species now persists in these 3 remnant populations at Dana Point, South San Mateo, and Santa Margarita. And the 3 remnant populations are small, isolated, and genetically differentiated. So recovery of the Pacific pocket mouse calls for conservation of these remnant populations, as well as reintroductions of Pacific pocket mice into unoccupied regions of their historic range. So in order to facilitate these reintroductions without negatively harming the remnant populations by taking mice out of those populations, a conservation breeding program was started at the San Diego Zoo Wildlife Alliance, led by Deborah Shire. And the breeding program was started to provide a source of individuals for the reintroduction. So the breeding program was founded by 10 to 20 founders from each of those remnant populations, which were brought into the ex situ environment and allowed to admix for more than a decade of the program. So in addition to providing a source of individuals for reintroductions, the breeding program also provides a unique opportunity to understand the fitness trade-offs of keeping remnant populations isolated versus outbreeding them. The breeding program provides data on survival and reproduction across a well-tracked pedigree, and we combined this with whole genome sequencing of about 500 individuals from the conservation breeding program and from the wild populations. So because our sample size ambitions were larger than our budget, we had to be really strategic about how we sequenced our samples. So what we did was we sequenced the founders of the breeding program at higher coverage, around 13x, and then we sequenced all of the descendants in the breeding program at lower coverage, about 1x on average. We then imputed genotypes across the pedigree using a program called AlphaPeel, which apparently is really commonly used in livestock, but isn't often used in conservation breeding programs. So we are really excited to apply this technique or this method to the breeding program. So AlphaPeel estimates shared chromosomal segments between parents and offspring and aggregates genomic data across generations to call genotypes. And in test samples that we sequenced at high coverage and imputed from low coverage, correlations between genotype dosage was high, and estimates of genome-wide heterozygosity between imputed and high coverage genomes were also highly correlated. So the methods seem to work pretty well. So we first looked at demographic history, recent and past demographic history of the wild remnant populations. So we estimated effective population size in the recent past, which we can do with accuracy when we have many more than one genome, as I talked about in the last study. In this case, when we have lots of good resequencing data, we can use linkage disequilibrium-based methods to estimate recent effective population sizes. And all populations, all 3 remnant populations, show recent declines, and from what were historically much larger effective population sizes. And the 2 smallest populations, Dana Point and South San Mateo, had the lowest effective population sizes. Dana Point in the most recent generation had an effective population size of 12.6, and South San Mateo was 21.4, so pretty small. And patterns of genetic variation follow what we would expect based on these recent effective population sizes. Dana Point had the lowest levels of heterozygosity, highest levels of inbreeding coefficients, and highest levels of homozygous deleterious alleles. And in the plot on the bottom, I'm showing the distribution of heterozygosity across the genomes of the median individual in each population. And the bars, the colored bars, show runs of homozygosity. So you can see in the Dana Point population how much of the genome, close to half, is covered by these long runs of homozygosity, whereas the Santa Margarita population has much, much fewer of these runs of homozygosity, and South San Mateo something in between. And it's maybe not so surprising that Dana Point has such high levels of inbreeding, because it's really under pressure from urban encroachment. The mice at Dana Point are restricted to this headland here, which is now completely surrounded by housing. Caitlin Curry and Eric Funk from the Conservation Genetics Group at San Diego Zoo Wildlife Alliance have also generated some data from museum specimens that were collected in the 1930s, before urban development really got into full swing. And when we compare levels of heterozygosity in historical versus modern populations, or historical versus modern mice from Dana Point and Santa Margarita, we find that the modern Dana Point mice have much lower heterozygosity than they historically did, whereas the declines are somewhat smaller in the Santa Margarita population. So we know that genomic erosion is happening in these populations, that they're losing genetic diversity, having increased rates of inbreeding, and increased rates of homozygous genetic load. And when we brought these founders from the remnant populations into our conservation breeding program and admixed them, we find that the admixed population has the highest levels of heterozygosity, lowest levels of inbreeding, and homozygous genetic load. So that suggests that gene flow could help reverse the impacts of genomic erosion in the remnant populations. Poisson models of reproductive success showed that outbred mice that have higher heterozygosity, lower inbreeding coefficients, and lower homozygous deleterious genetic load had more babies in the breeding program. And these trends were also significant when we restricted the analysis just to the captive-born mice, so excluded the founders from the analysis. And outbred mice with lower inbreeding coefficients and lower genetic load also tend to live longer. So together, this suggests that genetic rescue could benefit the wild population. So if we can facilitate gene flow from them, then we could potentially boost fitness of these wild populations. However, one complication is that there is karyotype variation. So Marlis Hook and Anne Mizuraka, also at San Diego Zoo, have been generating karyotypes for Pacific pocket mice for a number of years now, and they've found these two common karyotypes. One that represents the ancestral karyotype has a chromosome number, a diploid number of 2n = 56, and that karyotype is most commonly observed in the Santa Margarita and South San Mateo populations, and then a derived karyotype with one extra pair of chromosomes and a diploid number of 2n = 58 that was commonly seen in Dana Point. However, karyotyping is really tricky, and it's prone, especially in these pocket mice, to artifacts, and so it was really difficult to pin down whether these chromosomal differences were actually fixed across populations. So in order to identify the source of the karyotype variation, we teamed up with DNA Zoo to generate some high-C data. So high-C data provides information on long-range 2D and 3D structure of DNA molecules. So here are two contact maps. The one on the left is from a South San Mateo individual with the 2n = 56 karyotype, and then the one on the right from the Dana Point mouse with 2n = 58 karyotype. Each of these blocks along the diagonal, the red blocks, represent a contiguous chromosome with lots of contacts within that molecule. So when we compare these two contact maps, we find that one of the major differences between them is that in the Dana point individual, chromosome 2 is actually split into 2, suggesting that the extra pair of chromosomes is caused by a chromosomal fission or splitting of that chromosome 2. And when we zoom in on the chromosome 2 breakpoint and look at the SNPs that flank the breakpoint, we find that those SNPs are really highly differentiated between Dana point and the other two populations. And running a PC of those SNPs, a principal components analysis, clusters mice into these 3 clusters by their karyotypes. So when we run this PCA across all of our 500 or so mice, even those with an unknown karyotype, we find that all of the Dana point mice cluster into this homozygous fission karyotype, or the derived 2n = 58 karyotype, whereas all of the Sal San Mateo and Santa Margarita mice cluster into the homozygous ancestral 2n = 56 karyotype. And the only time we observe a heterozygous or intermediate karyotype is in admixed mice from the conservation breeding program. So that strongly suggests that these chromosomal differences are fixed in between the wild populations. And the reason that fixed chromosomal differences are a red flag for conducting genetic rescue is because they can reduce fertility in heterozygotes. So say we have a Santa Margarita mouse on blue in the left with that's homozygous for the intact chromosome 2, and we cross it with a Dana point mouse that is homozygous for the split chromosome 2. The offspring will be heterozygous, so have 1 intact and 1 split chromosome 2. And when the chromosomes properly segregate during meiosis, then viable gametes are produced, but improper segregation can lead to aneuploid gametes, which are inviable. So you can get a fraction of inviable gametes produced during every meiosis. So in the breeding program, mice with heterozygous karyotypes do produce offspring over multiple generations. And starting in the F2 generation and beyond, we actually start to see recombination of the ancestral and the fission haplotypes on chromosome 2. So in this heat map on the right, if you look at the F2 individuals and the individuals are rows, you can see that that the genotype associated with ancestral or fission will flip as you move along the chromosome. So that suggests that recombination is happening, homologous chromosomes are properly pairing and segregating during meiosis, but there still may be a portion of gametes that are inviable, which would result potentially in a reproductive cost to the heterozygous karyotype. So on the one hand, we know that mice with higher genetic diversity and lower inbreeding and genetic load have higher fitness, but there may also be this reproductive cost to the heterozygous karyotype. So then the question is, which is worse, inbreeding or outbreeding? So to get at this question, we ran multivariate models with all of our variables, first summarizing genetic variation, or first summarizing all of our highly correlated sort of genetic diversity variables by principal components. So those included metrics of heterozygosity, runs of homozygosity, and genetic load, and then running those together with karyotype in these fitness models. So we found again that mice with higher levels of heterozygosity and lower levels of inbreeding and genetic load tended to have more babies in the breeding program. But it was only when we accounted for this boost in overall genetic diversity did we actually start to see the potential cost of the heterozygous karyotype on reproductive fitness relative to the homozygous karyotype. But because mice that have this heterozygous karyotype also tend to have high heterozygosity genome-wide and low inbreeding and load. The cost of having that mixed karyotype is masked by that benefit of overall genetic diversity. So we can use this model to help us estimate the number of offspring we would expect when we keep a population isolated versus admix them. So for an isolated Dana point population that has really low genetic diversity, but also has this homozygous karyotype, we expect the average mouse to have less than one offspring. But with a boost in genetic diversity that comes from crossing Dana point with the other two populations, then we expect that the average admixed mouse, even with a heterozygous karyotype, is expected to have about 2 offspring, suggesting that the benefit of outbreeding outweighs the cost of the chromosomal differences. And for the South San Mateo population, which also showed genomic erosion, so elevated levels of inbreeding and loss of heterozygosity, we can cross it with a Santa Margarita mouse, which has the same karyotype, to boost fitness. And there may even be a benefit to admixing the Santa Margarita population. So in conclusion, we present a case here with the Pacific pocket mouse where, despite chromosomal differences, extinction risk is still higher if these populations remain isolated. And this case also demonstrates how empirical genomic data can help us weigh the costs and benefits of genetic rescue and apply it even in cases where outbreeding is a risk. And being able to apply these less restrictive criteria unlocks a larger toolkit that is available to us to combat the conservation crisis. Okay, so my last example is one where we use genomics to predict the viability of a future restored northern white rhino population. So just some background on the white rhino. It consists of 2 subspecies, a northern white rhino shown in orange and the southern white rhino shown in blue here. So swift conservation actions in the early 1900s allowed the southern white rhino to recover from about 50 to 200 individuals to more than 10,000 today. But the northern white rhino has followed the opposite trajectory and has declined over the last half century to now only 2 non-reproductive females are alive today. So the future of the northern white rhino depends now on cryopreserved cell lines from 12 individuals that are banked in the San Diego Zoo's frozen zoo. So the hope is that in the not too distant future, we can use these cryopreserved fibroblast cell lines, which are skin cells, reprogram them into induced pluripotent stem cells, or iPSCs, which can differentiate into any cell type. And we can direct differentiation to germ cells, which can make sperm and eggs, then use these gametes for in vitro fertilization to make an embryo that would then be transferred to a southern white rhino surrogate. So we've already been able to reprogram stem cells and direct differentiation to certain cell types. So this image here is cardiac cells that were differentiated from stem cells by Marisa Carodi at San Diego Zoo and her collaborators. So these cardiac cells will actually beat in culture, which is pretty awesome. And there's another group that's also working on restoration of the northern white rhino through cloning. And has already begun transferring embryos. So a restored northern white rhino population may be a reality in the not too distant future. So then a looming question is, will a northern white rhino population that's restored from these 12 cryopreserved cell lines be viable? And I just want to point out here that during this part of the talk, I have tried to highlight the northern white rhino in orange and southern white rhino in blue, because I know it can be like really difficult to keep the two tracked. So just think orange is the at-risk population, whereas blue is the population that's doing a lot better. Okay, so will a northern white rhino population that's restored from 12 cryopreserved genomes be viable? So to get at this question, we measured genetic diversity and genetic load in those cell lines that are banked in the frozen zoo and compared them to those of southern white rhino genomes for individuals that are alive today. And then we simulated fitness of a restored population in the near future. So a previous study done by Tunstall et al. Found that these cryopreserved cell lines actually have more genetic diversity in them than the southern white rhino population that exists today. So that suggests that there is adequate genetic diversity for this population. But a second question is, what about genetic load? So you'll remember genetic load is just the sum total of all of the deleterious mutations in the genome. And the reason genetic load is important is because it causes inbreeding depression. So inbreeding depression is reduced fitness of closely related of offspring of closely related individuals or of inbred individuals. And it's caused by deleterious mutations that were heterozygous in the parents that become homozygous in the inbred offspring, where they are expressed and affect fitness. And populations that have inbreeding depression are more likely to go extinct than a population of the same size that is not experiencing inbreeding depression. So this is an important part of, conserving small populations, is considering inbreeding depression. So we looked at genetic load in our cryopreserved cell lines and compared those to southern white rhinos. So again, here the northern white rhino is in orange, southern white rhino is in blue. And on the left side of this plot are homozygous deleterious alleles, and on the right are heterozygous deleterious alleles. So we find that the southern white rhinos have higher levels of homozygous deleterious alleles, and these homozygous alleles are more likely to be affecting fitness in the organism now. But the northern white rhino has higher levels of heterozygous alleles. So these are less likely to impact fitness in the individual now, but may affect fitness when they're converted into a homozygous state in offspring. So we can be fairly certain that a population founded by 12 individuals will have increased levels of homozygosity, including these deleterious alleles. So we can expect homozygous genetic load will increase in a restored northern white rhino population. So can the population avoid severe inbreeding depression? For southern white rhinos, that population was able to recover, and inbreeding depression did not markedly hinder their recovery. So we can use southern white rhinos as a benchmark for a viable population that can mount a recovery. So we simulated genetic load into the future in a recovering northern white rhino population using that southern white rhino population as our benchmark. So to do this, we took all of those variants that were identified in the cryopreserved cell lines, and we assigned selection and dominance coefficients to deleterious variants in the genome. We then simulated 10 generations of non-Wright Fisher evolution under scenarios of different levels of population growth, ranging from no growth to 50% per generation growth. So here's just a diagram of what we did starting in generation 0. We actually started with our 8 unrelated northern white rhino genomes. 4 of them were or related to others in the population. Then, started a population in generation 1, and then simulated all the way to generation 10 under different growth rates, and each generation measured genetic load and fitness. So we found that rapid growth of the population would be needed in order to maintain fitness levels that were, comparable to or better than the southern white rhino population. So in all scenarios, we found that homozygous genetic load, which is in the plot on the left, increased over time. And the mean level of genetic load for the southern white rhino is shown in the hatched line. And then the same with fitness over time. Fitness declined as we would expect. And here again, the southern white rhino line is plotted, or the southern white rhino mean is plotted with the hatched line. And when the population grew by less than 20 to 30% per generation, then genetic levels of genetic load exceeded that of northern white rhino, and fitness dropped below. So that suggests that that growth of 20 to 30% at least per generation is required to maintain fitness comparable to southern white rhino. But the beauty of having cells for population restoration is that we can repeatedly reintroduce banked genetic diversity back into the population as it's lost over time. So that's what we simulated next. We did the same thing as before, but this time we took each generation one randomly selected cryopreserved cell line and reintroduced it back into the population. And we found that reintroduction of our founder genomes helps to avoid inbreeding depression and effectively releases the population from the requirement of rapid growth to avoid severe inbreeding depression. So in all cases, genetic load remained below that of the southern white rhino, and fitness remained above that of the southern white rhino. So that suggests that inbreeding depression from high genetic load is not necessarily a barrier to recovery of northern white rhinos, because we have this, nearly infinite resource of these cell lines. So in conclusion, a restored northern white rhino population can avoid inbreeding depression, and that's mainly because restoration from biobanked cells is not subject to the same constraints as traditional restoration from a really small set of founders. Okay, so zooming way out on all of these three cases, today I presented these three cases where we use genomics to help us assess conservation status across species using reference genomes, use population genomics to guide strategies to conserve endangered species, and use genomics and simulations to predict the future viability of populations. But there are also lots of other applications that we use genomics for in our lab and in other labs at the zoo and other collaborating labs, including dietary reconstruction with metabarcoding, the use of portable sequencing from Oxford Nanopore for generating genomic data in the field, using amplicon sequencing to identify disease status, RNA sequencing to understand gene expression, and that's a big one for the northern white rhino project, and increasingly using methylation to understand aging and stress in populations. So there's a lot of applications that we can use genomics for, and I think it's a really exciting time to be in this field. So with that, I just want to acknowledge collaborators on all of these studies and my funders, and thank you very much for your attention, and I'm happy to take questions.
Thank you so much, Dr. Wilder, for such an interesting presentation. I'm going to open it up now to any questions that anybody has in the room or online.
I have, but there is one.
Lael, if you want to ask.
Hi there. Can you hear me?
Yeah, I can.
Great. Thanks for the really interesting talk. I wanted to ask about heterozygosity because my impression was that it looked like heterozygosity was quite a good predictor relatively for conservation status or population health. And I wondered if you could comment on heterozygosity as kind of if that, to what extent it can be used as a population independent kind of absolute metric, or whether you really have to calibrate your heterozygosity thresholds for different populations.
Yeah, heterozygosity is tricky because, on the one hand, um, like I talked about in the zoonomia analysis, so using reference genomes to understand extinction risk, it can tell you something about how large a population has been historically. So, large populations that were, historically large are more likely to be also large today, and large populations are less likely to go extinct. But the sort of double-edged sword of it is a population that does have really high heterozygosity can also have really high inbreeding load. So lots of these deleterious mutations, like the northern white rhino had, that are in a heterozygous state. And when that population contracts, so if it undergoes a really rapid population decline, then you can get severe inbreeding depression. And so there is a lot of sort of nuance to how we use heterozygosity to understand extinction risk. When we're looking, if we look at a population that we know nothing about, and we see that it is really heterozygous, then we might think like, okay, this population we at least know used to be large and, is probably still large today, or, is more likely to be large today. But if we see this population is really heterozygous, but we also know now it's really small, it actually could be at an even higher risk of severe inbreeding depression. So that's sort of a complex idea, I guess, but kind of demonstrates how heterozygosity is not just kind of a simple metric to use. Does that answer your question?
Yeah, awesome. Thank you.
Hey, first of all, congratulations, Dr. Wilder, for the nice talk. I could talk to you for the whole afternoon, but I'm gonna pick just a few questions. So, how do you identify the deleterious mutations, deleterious alleles in the genome?
Yeah, so it has been in all of the cases that I present, it was always bioinformatically. So either using predicted changes to protein coding genes. So where we would assume that a non-synonymous mutation is more likely to be harmful than a synonymous mutation. Something like a loss of function mutation, like a premature stop codon or frame shift mutation, is more likely to be severely harmful or to have a really high impact. And then, like I mentioned before, if we look across species at a particular site in the genome and we find that it's really highly conserved across species, then we would expect that that site is really important and it's conserved and under strong purifying selection, and any changes at that site are likely to be deleterious. So it was a combination of all of those things. So that said, that's, it's all sort of a bioinformatic way of assessing genetic load, and there were no actual experiments done to validate any of this. It was all just kind of like, counting up these putatively deleterious alleles in the genome. But, using this approach of phylogenetic conservation has actually identified a lot of known disease variants in humans. And so, these approaches in general have been validated, but there were no particular mutations that were actually validated in any of these studies with direct evidence.
And if I can ask you one more question, especially in that rodent population you have studied through the years, have you assessed the new de novo mutations in that population? How frequently it happens?
No, I've not looked at that at all. Yeah, I mean, we have. Are you talking specifically like in the breeding program?
Yeah. I guess you are measuring the genetic diversity in that populations under recovery.
Mm-hmm.
And I would like to know if you have seen de novo mutations and how frequently are they in this recovering population?
Got it. I haven't looked at that specifically. My guess is that it would be pretty infrequent. I mean, we've got, 10 generations or so. And so, it would certainly be a small contributor to overall genetic diversity. But yeah, I haven't looked at that specifically. We do have that kind of data for some individuals, where we have parent and offspring sequenced at high coverage. So it would be interesting to look at that.
Thank you.
Yeah, thank you.
Quickly, just to finish this up, Shelby, do you want to ask the question?
Yeah, it's really quick. I was just wondering, why are herbivores more susceptible to a small effective population size compared to carnivores?
So the short answer is I don't exactly know, but there was a previous study published that found that herbivores were more likely to be threatened with extinction at higher population sizes, so census population sizes. So it may just be that they have these larger census population sizes kind of at equilibrium than a carnivore population does, and maybe are susceptible to population fluctuations that are at a larger size. So yeah, I'm not exactly sure, but I know it is probably correlated with whatever dynamics are happening at the census population level.
Thank you.
Thank you so much. With that, I think we've run out of time. We've gone over time, actually. So thank you, everyone, for such a great discussion. And thank you again, Dr. Wilder, for presenting to us today. The recording will be posted on YouTube, and we can send you those details later. But everyone, thank you so much for coming, and we'll see you next week.
Hey, thanks, everyone. It was a great discussion.